Hello, this is take two for this PowerPoint practice number 3 for this Unit 5. Okay, so once again, take a look at this question, scratch down an answer, flip the slide, and I'll walk you through it. Okay, so here's the pictures, and they're asking you, if this cell does meiosis, what do we get? They describe what this cell is, and these chromosomes and the genes that they want you to draw here. Make sure you use their symbols. Don't make up your own symbols. If they tell you that we have G's and D's here, right? And they tell you what chromosomes they're on. The G's are on chromosome number 1. They tell you that this F1 individual that this cell belongs to, right, has a big G that it got from this parental homozygous dominant, and a little g that it got from this parent, right? And a big D and a little d likewise. So it's your classic F1, right? Heterozygous for both pairs of genes, right? And they expect you to know that that means, since we told you that G's are on chromosome 1, that one of these chromosomes has 2 big G's. After replication makes 2 chromatids. One has 2 little g's, and likewise on the chromosome number 2, 1 chromatid with 2 big D's and 1 with 2 little d's. So even if crossing over happens here, that doesn't really matter. You can only make 4 different kinds of gametes, and you need to now correctly have all of the following: that in your little drawing, you don't have double-stranded chromosomes. They must be unduplicated, right? And you have to have the G's on chromosome number 1 because they told you that right here, right? And then you have to have these 4 combinations, right? And so you got to get all that right to get that 1 point, which is not hard at all, okay? But you got to get all that stuff right. Okay, next one, take a look, scratch and answer. Here we go. Okay, so now they ask you predict the possible phenotypes. So now they don't want G's and D's, right? You got to take that into account to figure out your answer here, but they want you to talk about green flowers or purple flowers or tall or dwarf, right? So that, understanding that first, we have our F1 that we just had, right? Heterozygous for both. And now we're going to cross it with an individual that's homozygous recessive, your classic kind of test cross, right? So this individual makes up only one kind of gamete, right? All of them have little g, little d. And this individual we just showed can make 4 kinds of gametes. In part A, that's basically what you just showed. Gametes with 2 dominant genes would make this kind of kid when combined with the gamete from this parent, right? And then we have, right, this kind of possibility and this kind of possibility and this kind of possibility. Right, so you do your Punnett square, you come up to these 4, but again, that's not what they want. They want phenotypes. So you simply have to convert that to phenotypes. And what else did they ask you for? Predict the possible phenotypes and their ratios. They didn't bold the word "and" here for you, but you need to have some numbers here. So 25% of each would have sufficed. You could do it in proportion form, right? 1 to 1 to 1 to 1. 1/4, 1/4, 1/4, 1/4, but you have to have both numbers and phenotypes, not genotypes, to get that 1 point. Not hard, but you could trip up in one of those 2 ways. Next and last one. Okay, so here we go with this one, right? If now these 2 genes are linked, they're on the same chromosome, right? How would they differ from that 25, 25, 25, 25% that you just showed with that test cross of F1 and that GGG individual? Well, if these genes are on the same chromosome, then this F1 individual from this dad got a chromosome that had, or mom or dad, whatever, got the two dominant genes linked. And then from its other parent, it got a chromosome with the two recessive genes linked, right? And so, right, they could only give their kids this chromosome or this chromosome. So if they gave it this chromosome, you'd have a kid that had that genotype and showed both dominant phenotypes, right? Because they're also asking you about phenotypes here, not genotypes, right? Or the kid that got this chromosome in a gamete, along with what it got from that individual, had both recessive traits, okay? Remember, in the parents where they were unlinked, right, the genes were unlinked, you'd expect 25% of these, 25% of these, and then 25% of those other kinds, which you wouldn't get here unless crossing over happened, which it's going to. But really, you didn't have to add that into the equation here. If you simply said they'll have more than 25% of these kind, you would have got that 1 point. Or if you combine those and say, well, if they were unlinked, you'd get 50% of this kind or this kind, but now you'll get more than 50%, you would have been okay. Or if you said, okay, those kind of kids that could only come about by crossing over, the recombinant kids, right? That somehow these genes, by crossing over, these switch places, so you could get kids like this, but I wouldn't predict that you would get 25%. I'd predict less than 25%. Any one of those answers would have given you that 1 point. So now, here's this next one, right? Both mitosis and meiosis produce daughter cells containing genetic information, two events that are common to both that ensure that the daughter cells get the right number of chromosomes. Well, in mitosis, the right number is the same amount as the parent cell, and in meiosis, it's half. What two events that are common to both make sure they get those right numbers? Well, it's the separation events, right? It's the separation events. It's the lining up before they're separated events. But a key event is that copies are made before either meiosis or mitosis. Then dividing those cells into two or maybe four, right, also is part of making sure that they get the right number of chromosomes. So are these other things, right, which they added to this list of 7 different things you could say to only get 2. Now, remember, they told you to describe 2 events. If you described all 7, they're only going to grade the first 2. Well, if you describe these 7, you'd be okay because you get those 2 points. But if you describe something that wasn't on this list and you describe 5 different things and you describe that thing first that wasn't on the list, unfortunately you're not. So pay attention to, again, when they ask you to do 2, 3, 4 things, only do that many things. And if you can think of several things, pick the things that you think are most important. Okay, so now, they now tell you that the genetic composition of daughter cells is different if mitosis happens compared to meiosis, right? Describe 2 features of the cell division process that lead to these differences. Well, 2 features of the cell division process, right, are you get 2 cells here, you get 4 cells here. Because here you only have 1 division, here you have 2 divisions. And that's why the genetic composition is such a difference. Here you have 1 set of chromosomes when it's done. Here you have 2 if you start with a diploid cell, right? So this is the feature of the cell division process that results in that 1 set as opposed to 2. So that's one thing you could say. But notice that if you said both of these things, you can only get 1 point. So pick two different topics. Again, you're asked to describe two features, right? How many is two? Let's pretend this is, kindergarten. How many is two? Hold up your fingers. Two, right? Did you just hold up your fingers? Okay, that'll be our little secret because we can't see you do this. All right, so anyway, um, what is also a feature of the cell division process? Well, here, just saying that it results in diploid cells or haploid, like we just said, that's okay also. That's a feature of the process. It's not describing something about the process, but it's describing the end result of the process, okay? Here we have that um, chromatids separate in mitosis, but they only separate in meiosis II here. So that's another thing you could have said. Crossing over does not occur. Crossing over does occur. What difference does that make to the genetic composition? That makes all of these chromosomes the same, or excuse me, all the genetic composition of the daughter cells the same after mitosis. It makes them all different after meiosis. This is the thing that happens, and this, that make that happen. All cells different or all cells the same. And the fact that here you have homologous chromosomes not separating from each other, therefore both sets of chromosomes remain in the cells that you get, the daughter cells. Here it does occur, you have different cells. Any of those two things. But again, if you describe 800 different things, they're only going to grade the first two. So this is about as true as it can be. And I'm getting ready to exercise myself. I make up my own exercises. I go in the ocean and do exercises. And I got one that I do on the beach actually where it's like a hybrid between a lunge and a crunch. And I'm about to go do those now. You know what I call that hybrid between a lunge and a crunch? I call it lunch. It's time for lunch. I hope you liked all that.